Hello friends, today we will see some concepts of qualitative analysis. Some concepts of qualitative analysis. Then what is qualitative analysis? It is nothing but what is present in the given sample. Means suppose if we take inorganic sample, then some cations and anions like Mg2 plus Cl minus etc. may be present in the sample, and we are going to find it out by some chemical means. And what is quantitative analysis? That is how much is present in a given sample. Suppose Mg2 plus is there in the sample, then we have to find out its concentration. It comes under quantitative analysis. Then here we are only focused on concepts of qualitative analysis. Depending on quantity available for the given sample, we classify the qualitative analysis into 5 categories. First category, macro analysis. Here, the amount of substance taken of the sample, if the substance is in solid state, then it's 0.5 gram to 1 gram. And suppose the sample is in liquid state, then it is of 20 to 30 centimeter cube. If you see, the amount of substance is quite larger, and we can perform its analysis so easily. Second, semi-micro qualitative analysis. The amount of sample in solid state is of 0.05 gram to 0.1 gram, and if it is in liquid state, then it is of 10 centimeter cube. We perform it in our undergraduate practicals for both organic and inorganic semi-micro qualitative analysis. Third, micro analysis. Here, the amount of substance of the sample is of 0.01 gram to 0.05 gram. Here, we perform some spectroscopic analysis to detect the amount of substance present. And if suppose Mg2 plus or K plus or Na plus, if present in this much amount, then we go for flame photometry. And it is rather difficult to analyze in a laboratory by some chemical means or by some chemical tests. Fourth one, ultra micro analysis. Ultra micro qualitative analysis. Here the amount of substance of the sample is of 1 milligram, and rather it is quite difficult to analyze in the laboratory by some chemical test, and here also we go for some spectroscopic technique. And the last one, fifth one, is stress analysis, the amount of substance is very less than 1 milligram. I repeat, very much less than 1 milligram. And it is quite difficult to analyze in the laboratory again by some chemical mean, and we go for some advanced technique to find it out. We will focus on semi-micro analysis. Now we will focus on semi-micro qualitative analysis, as we have it in a syllabus for the practical for undergraduate students. For it, we take amount of substance of 0.05 to 0.1 gram. If we talk on semi-micro qualitative analysis for inorganic substances, then the salt provided is of this much grams. What we perform there is, if we take the mixture in a dry test tube, some charcoal test, flame test, and sometimes even if we add concentrated HCl, H2SO4, etc., and we get some colorful solution. Even some gases are also evolved, and from that we detect that particular cation or anion is present in the given sample. If we have to find out Cl minus, then we add AgNO3 as a precipitating agent, and we get white color precipitate AgCl. Now we will focus on gaseous evolved during the reaction. If salt contains ammonium ion, then definitely on heating it will give out ammonia gas, and this ammonia gas can be detected using moist red litmus paper. It will turn it into blue, which indicates NH4 plus may be present. Secondly, we can use turmeric paper, moist turmeric paper, and if suppose it turns it into brown, reddish brown, then we can conclude that in our substance NH4 plus is present. Sometimes even carbon dioxide gets released. For example, if we heat calcium carbonate, it will release carbon dioxide, and this can be detected using lime. It turns it into milky, which indicates carbonate ions present. Third, sometimes gas like sulfur dioxide gets released. Here it can be detected using potassium dichromate paper, which we have to acidify by H2SO4. If potassium dichromate paper turns yellow, then we can conclude that some sulfide ion may be present. Then we have talked about gases evolved during particular reaction, performing it in an experiment of semi-micro qualitative analysis. In semi-micro qualitative analysis for an inorganic compound, sometimes even we use some papers to analyze the cation or anion. Now we will see a roll of papers impregnated with some reagents. And first we will see starch iodide paper, which are easily available in a market, or we can make it in our laboratory using starch and Ki. We mix it, we soak it in a water, and definitely we have to add some strips of filter paper, dry it, and it can be utilized as a starch iodide paper. Then here we can easily analyze some anions like Cl minus, Br minus and I minus. If my compound contains Cr minus, on heating it, that substance, it will release Cl2 gas, which can be detected using starch iodide paper. That paper will turn into blue, which indicates Cl minus is present. And if suppose for Br minus, the starch iodide paper will turn it into brown, indicate Br minus is present. Similarly for I minus, it will turn into again blue, indicating I minus is present. Second paper we use is potassium dichromate, that is K2Cr2O7. What we have to do? Just take K2Cr2O7 in a water, dissolve it, and take few strips of filter paper, then soak it, and after drying, it can be utilized as a dichromate paper. What we can do? We can analyze sulfide ion using it. Sulfide ion, if my sample contains sulfide, then heat it, then the gas will evolve, which on putting potassium dichromate paper, which is already acidified with H2SO4, here we can detect whether sulfide ion is present or not. What we have to do? Just take the substance, heat it in presence of some acid, gas will evolve, SO2 gas will evolve. On putting potassium dichromate paper, which is already acidified by H2SO4, that will turn it into yellow, indicating sulfide ion is present in my sample. Third paper is lead acetate. Here we can detect S2 minus. so easily. Lead acetate paper can be prepared by preparing a solution of lead acetate, putting inside few strips of filter paper, soaked and dried, and it can be utilized as a lead acetate paper. Then here we can detect S2 minus present in my sample or not. Just take the sample. In presence of H2SO4, it will produce H2S gas. While evolving this gas, we can put over the mouth of the test tube our lead acetate paper. If this lead acetate paper turns black, producing PBS, indicating sulfide ion is present in my sample. Fourth, we are going to use is DMG, dimethyl glyoxam. Take dimethyl glyoxam in an ethanol. It will get easily dissolved in it. Put few strips of filter paper for soaking, and then on drying that papers we can utilize it as a DMG paper. Then it is most important to detect few ions like nickel. Nickel reacts with DMG to give nickel DMG complex, which is scarlet red in color, indicating nickel is present in my sample. And lastly, we will see oxime paper. What we have to do? Just take 8-hydroxyquinoline, if you want to detect Mg2 plus, then nitrogen and oxygen is having lone pair of electron. It can donate to the magnesium, and it can be easily donated to magnesium, and a complex is formed, magnesium oxynate complex, which is yellow in color. That oxime paper gets yellow, indicating magnesium is present in our sample. Then here we have seen that, see how this different types of paper impregnated with some reagent can be useful to detect anions and few cations. And we take it in a very much small, that is semi-micro amount. And at last, I request you to like my video, to share my video, and to subscribe my video. Okay? Have a good day. Thank you.